Sci-Bono Discovery Center. My name is Tolani Nameka, and I'll be the program director for this program that we came for. Our CEO that have joined us, the guest speakers that have joined us in this room, friends and colleagues that have joined us online, you are all welcome to this talk. Right, we requested the educators to bring you to our Speak to a Scientist program, and I will just say a few words about this program. What is Speak to a Scientist? Speak to a Scientist is a platform whose objective is to provide an opportunity to clarify thoughts, generate conclusions, develop theories through asking questions differently, and to expose our learners to new ideas and perspectives, thereby encouraging growth of new understanding. But because science has proven to be a benefit to human life, giving us an opportunity to establish norms that ease new learners' inhibition, motivate sharing and promote respectful communication. It is in this regard that we like as well to invite you learners that we are here today for you to be exposed to a cognitive demand on that from the monologic convention type of education that we are doing in classrooms to more interaction that is intended in building on your thinking, which requires more elaboration, revisiting ideas, and clarification of meaning that will be witnessed today from our guest speaker that we have invited. It is with this repute that as a science center, our vision sits precisely on the idea of new learner engagement, which gave rise to this program, Speak to a Scientist, to practically demonstrate via various modes of communication why science is central to everyday life in terms that even a lay person can understand. Starting with why what we do, we deem it relevant. Without any waste of time, ladies and gentlemen, please allow me to call upon our Senior Science Center Manager, Mr. Akash Dasrath, to come and introduce our guest speaker. Let's give him a hand.
Thank you very much, Program Director. I think I'd like to start off by welcoming you all to the Speak to a Scientist event. It's so good to see all of the learners here, the educators here, and of course our very special guest and her team, Professor Himla Soodyall. Together with that, I'd also like to welcome our CEO that's joined us today, Professor Nsobi, and our acting COO, Mr. Anele Davids, amongst other managers that are also here today. Just to start off, I want to give you a little bit of background of our guest speaker today. Himla Soodyall is the Executive Officer of the Academy of Science of South Africa, which is ASEF. She is a research professor in human genetics at the University of Witwatersrand and was principal medical scientist at the National Health Laboratory Service. Soodyall has done groundbreaking genetic research into the peoples of sub-Saharan Africa, in which her studies have identified some of the oldest DNA found in living people today, adding weight to the theory that modern humans evolved in the area now known as Southern Africa. Her talk today is entitled Genetics and Society, and I hope you guys will enjoy it and will have lots and lots of questions at the end to ask her. And so without further ado, I'm going to hand over to Professor Soodyall. Let's give her a big round.
Wow, look at the audience. It's so nice to see all you enthusiastic people sitting there with your beautiful uniforms. You really, really look wonderful. Well, good morning all. Thank you, Program Director, the CEO and colleagues and members of Sci-Bono, and of course my team. It is indeed a pleasure for me to be here today. It's been an item on our list of activities to do for quite a long time. So as you've heard, for the past 30 plus years, I've been working at the bench in a laboratory doing genetic research. When I started off, I knew I wanted to become a scientist, but I didn't really know which area of science I wanted to get into. So I started off my career at the University of Durban-Westville in KZN, and I took microbiology and biochemistry as my subjects to qualify for my undergraduate degree. And one day while I was in the library and I was reading some science magazines, I saw a very exciting article about spillage of oil on the ocean, and that scientists were thinking of creating, using what we call recombinant DNA technology, making a superbug that could have the elements in this bacterium that would break down the crude oil. And this was fascinating, because in the mid-80s, there was this new field of recombinant DNA technology. Up until that time, genetics was studying family history, using blood groups, blood group markers to study the genetic relationships of people. But now, with the impact of DNA, you remember who discovered DNA? The structure of DNA, anybody remembers? Yes?
James Watson.
Yes, well done, well done. So now we're going to go into the story of genetics and heredity. And as our colleague up at the top identified, in 1953, almost 70 years ago, Jim Watson and Francis Crick identified the structure of DNA. Now, in that time, physics was a very important field. All major scientific discoveries were done with physics. And at this stage, people were like trying to decide. They knew that there was a biological element in our cells. And somebody named it DNA many years before, but they didn't know what it did, how it looked. And these two guys discovered the structure. And Jim Watson was very, very young at the time. And if some of you have the opportunity to find the video telling the story of Jim Watson, you will really, really enjoy it. And I had the privilege in 2000 to host Jim Watson in South Africa. That's him in the middle. My boss at the time, Professor Trevor Jenkins, who taught me the genetics that I know today. And here's me, much, much, much younger, in the presence of two iconic scientists. And you know, when you're young and you're enthusiastic and you meet these bigwigs people, you go, oh, wow. Anyway, Jim Watson had written to my boss, Trevor Jenkins, and the late Professor Tobias, who was the paleontology expert in the country. And he said he wants to come to Africa to trace his roots. He had heard me speak at Cold Spring Harbor Laboratory in the US previously when I was talking about my work on the peoples of Africa, and in particularly the people we presently call San and the Khoi. And he had seen what work I had done, and he was very intrigued by the stories that I was telling them at this big conference in the US. And then he wrote, and he said he wanted to come to Africa. You know, when Americans talk about South Africa or any place in Africa, they just say Africa. And if you ask them where's South Africa, they say they don't know. Okay? So, but Prof. Watson knew where South Africa was because he'd hosted us previously. And he wanted to come to South Africa to meet some of the San communities. So one of our colleagues from Wits University, who was studying the language of one of the southern group of San languages, they took him into the field where his research site was, and for the first time Jim Watson stayed in a tent. I inherited his tent, he gave it to me. So that's the other story. But the best story was, we were in the Drakensberg, taking him to see rock art paintings. And we went to Game Pass, which is now a heritage site, part of the visits that you could do. And it was in April, so it had rained. And as we were walking to the mountains to see the rock art display, we had to cross a stream. But the stream was now flowing much heavier after the rain. And so it was difficult to pass. So the archaeological colleagues from Wits Rural Rock Art Center, they stood astride in the water because they had those heavy boots, and they helped us use their feet as stepping stones to go to the other side. Jim Watson now, because he's tall and he thought it would be too sissified if he had to do this, he decided to jump. Now it's a big wide thing. Without much of a running start, he decided to jump. As he reached the other side, he slipped, he fell, and all we could hear was, "Dum!" His pants were staring at the back. So this was, now we're like, oh wow, we're hosting one of the Nobel laureates. Remember, they got the Nobel Prize for the discovery of DNA, and here we are in the field with them, and he's just fallen, and torn his pants, and I had to walk all the way up to the mountain with his torn pants, and for the rest of the weekend, because that's the only pants he brought to go on the trip. So that's my famous story about Jim Watson, and what I'm saying is, you never know in your life who you will meet, and the impact that engagement will be. So congratulations to Sci-Bono for bringing in to their environment the Meet the Scientists. Because there may be one small little thing you pick up that will inspire you, and that's what I hope to do today. So that's a very long-winded introduction. So what I want to talk to you about today is, how is it that our species, have come to occupy the rest of the world? It's like the migration of humans. And my side of the story is about the use of DNA. So you all know where your DNA resides in your cell, correct? Yes. Which structures? No, you gave me the first answer. Somebody from this side? Come on, it's on the picture. Yes? The nucleus? Is the nucleus the only place where DNA is? Yes, next to you? Very good. So our DNA is located in two structures in the cell. The nucleus, as you correctly pointed out, and this DNA, we get half the number of chromosomes from dad and the other half from mom. Who can tell me how many chromosomes humans have? Yes? 46, 23 pairs. Now, the first 22 pairs we call, starts with an A, autosomes. And then the other pair are the sex chromosomes. Right, maritime school. How many X chromosomes do women have? You say 3, you're abnormal. 2, one from dad and one from mom. So with respect to the sex chromosomes, girls have 2 copies of X, one from dad and one from mom. What chromosomes do boys have? X and Y. Who do you get your Y chromosome from? Y? Y? You see, that small little chromosome there, that is what makes you boys boys, on your Y chromosome. It is so small, but sometimes boys and men think very big about themselves. So this is what we call the karyotype, the arrangement of the chromosomes, the 23 pairs, half from dad and half from mom. Are we all sharp about the nuclear DNA? Cool. Then the other structure is the mitochondrion. Now the mitochondrion has a very fascinating history. When biological specimens were all single cells, the mitochondria and one prokaryote and another one decided to play nicely together. And you say, come, let's live nicely together, symbiotically, and we'll share the work. My job will be, I'll make the energy, and you can do the other things. So through the symbiotic relationship, two bacterial, very primitive cells got together, and over time, with evolution, millions of years, they kept the mitochondrial DNA relic. That second DNA stayed in the cell to become the mitochondrial DNA. So even now, us all, we have DNA in two structures, but the mitochondrial DNA we only get from mom. As always, women are the powerhouse. So they make the energy. And all of us, girls and boys, get the mitochondria from the mom, but only the girl children will pass it on. Because we want to keep the power. Right? So we keep the power. So mitochondrial DNA is passed from mother to all children, but only the girl child will pass it. So from a genetic tool perspective, we can now use every single individual in this room, girls, boys, men, and women. We can study your mitochondrial DNA, and I can trace your mother's line, mothers, lineage all the way to the past. And the reason for that is that this structure does not reshuffle or mix. Remember that process of recombination you learned about with chromosomes? That doesn't happen with mitochondria. So we now have a very dynamic tool. In the absence of recombination, following exclusive mother's line, we can now track maternal or mother line history in human people living today. So the key of genetics is we study DNA in living people and we reconstruct to the past. Whereas our fossil colleagues, the paleontologists, they have to dig and find specimens. They study the structure, or what we call the morphology, and all kinds of other things, and then they reconstruct to the present. Archaeologists, on the other hand, they go and they dig at settlement patterns, they make a survey of the sequence, what foods people were eating, they look for seeds, they look for tools, they look for pottery, and so on. So in asking the question, where did humans evolve? How did we come to spread to populate the rest of the world? Depending on what question you ask, you will decide on which tool you're going to use. Is that clear? So as genetics, our tool is DNA. I can get DNA from a hair, the root, from your saliva. If you smoked a cigarette and you left your stompy, we can get DNA from that. So DNA is very, 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 very powerful. It does not lie. You can change your clothes, you can change how you look in one generation, but you can't change what you're carrying in your DNA. That is why for most cases today, people use DNA to track relationships, etc. You're all with me so far? Yes. So those were the key messages. The key messages are for you to understand where in the cell we have our DNA located, how we inherit the different types of DNA, nuclear DNA and mitochondrial DNA, why we use mitochondrial DNA to trace mother line or maternal ancestry, and so that you boys don't feel bad, we even use the Y chromosome to trace what you inherited on your Y chromosome from your father, your grandfather, your great-grandfather. You study that. Okay? So that's the inheritance and how we link it to biology of the cell. Now you would also come across another very famous monk by the name of Gregor Mendel. And he, you know, these monks, sometimes some of them make beer or wine, others plant stuff, because that's how you pass your time in the old days. So this monk used to grow peas. And as he was crossing different specimens of peas, he noticed some peas were smooth and some were a little bit crinkled. And just studying the crossing patterns, we learned from him Mendelian inheritance. Now you will learn about autosomal inheritance, X-related. I'm not going to complicate it because I'm sure you're going to see all of that in your classroom environment. Okay, so now we know about Mendelian inheritance, and those of you who want to become medical scientists or doctors, it's very important for you to understand this very basic concept. Because the first thing you do when you see a patient, other than being polite and making them feel comfortable and at ease, you get their family tree. You draw their family tree, the parents, them, their children, their siblings, and so on. Then you ask certain questions. So if somebody comes to you with a problem with their hearts, you will say, well, who in your family has a heart problem? You document all of this and you will draw it in a pedigree kind structure. And then you may take a blood sample, you may do any other test to help you to make the diagnosis. But it's very important to understand family trees, and I'm sure some of you have done that as exercises in the classroom. So from seeing how we inherit all our DNA, this slide is just showing you all the other people not represented in your mitochondrial inheritance. So we have 2 parents, 4 grandparents. How many great-grandparents we have? 8. In the next generation, how many ancestors we have? 16. And then? And then? And then? And then? Cool. You see where I'm going? Now think about this. If you go to 7 generations, the number of people who contributed to your genetic makeup is 2 to the power 7. But only one of those ancestors would have contributed to your mitochondrial DNA. You understand the difference now? We're only tracking one line. And if you're a male, only one person, one grandparental line would have contributed to your Y chromosome. So although mitochondrial DNA is very, very useful for us to trace ancestry, it doesn't tell us everything about us. So if I want to know what heart disease I have, I'm not going to study mitochondrial DNA. I have to go to the genetics or the DNA in the chromosomes. So, but we have exploited the properties of single-parent inheritance and the absence of mixing or reshuffling that we call recombination, and we have studied inheritance in populations. And so I'm going to show you a few slides, but before I forget, I just wanted to summarize that if we ask the questions related to the past, there are many, many tools that we can employ. Genetics, archaeology, anthropology. There's even a study of languages. Do you know that the languages we speak also belongs to families? And you can trace this by the use of certain words. So if you say, what is the word for cow in different languages? Then they will come up with it and then they'll say, well, this word is common, but these are variants or derivatives or things that are derived from that. So linguists have their own tools. Archaeologists have their tools. Paleontologists have their tools. Us geneticists have our tools. We all come from a different angle. We all work on certain assumptions. But at the end of the day, it's one history, right? So we will try to converge to that common point. And one aspect of genetics that allowed us to have a window much, much further into the past is ancient DNA. So we are living people, we are contemporary. So we can study our biology because we can take a blood sample, a cheek swab, a hair, you know, any skin tissue, whatever. Every cell in our body has the same DNA. But with ancient DNA, you can now they've been able to get DNA out of a Neanderthal specimen that lived over 40,000 years. And depending on that environment where that specimen was, the acidity, and so forth of the soil, some of that DNA is preserved. So the best source of DNA from ancient material, believe it or not, is the teeth. Because the tooth, in the middle of it, you have the dentine and all that pulp. So the specialists who work with DNA, they work like in spacesuits and all of that. Like, during COVID time, how people had to protect. You want to protect your sample so you don't contaminate it. And so fortunately, there's some technologies like the polymerase chain reaction. Have you heard of PCR? Maybe not. Maybe you'll hear it next year. But it's like a photocopy machine for DNA. You can make many copies. And so you can take small amounts of DNA and the target that you're interested in make a lot more. And so this was what has been used with ancient specimens, and people have been able to get DNA samples out of that, and then that has helped us to look further into the past. So I'm jumping the gun a little bit, but I also want to share with you another very important concept, and that is the concept of informed consent. Now, while I'm talking about this to you as a researcher, you must always question anybody who asks you for anything, particularly your sample. Informed consent means I must inform you about what I'm going to do with your sample, and if you have opportunities to ask questions, you must do that before you give consent, written consent, with a signature. Informed consent is the fundamental start of any research project. Even if somebody wants you to participate in a study about how much money you have to live in your household, they must explain why they want to do that study. You must give permission, and you enter into a contract with that person so that they don't sell your DNA, your information to everybody. So right now we have an act called Protection of Personal Information, or the POPI Act, to allow us to protect ourselves. Now you would have noticed when you came in the gates, the security person took some information, took the registration of the vehicle, he's doing his job. What happens to that information long term? Who has access to it? Because if you see the car license, it's got your registration number, it's got your VIN number, the engine number, all of those things. Where does it sit? How long does it sit there? And who has access to it? Similarly, when you're doing genetic studies, if you as a study participant volunteer to be part of my study, I can't give your samples and results to everybody, because then I'm breaching the contract I had with you for confidentiality. Confidentiality is a very important thing. So that's the other important factor I want you to take out of this talk. The issue of informed consent. You need to understand what you're getting into, what is going to be done with it. If you want to pull out of a study at any time, you have the right to. So you must understand your rights. And this is a lesson not only for genetics, it's a lesson for life. You must question stuff. Don't take things for granted. So informed consent is important. We have to do our work in fields and how you engage with the communities. You mustn't be what we call a parachute scientist. Fly into the place, take your samples and buzz off. No. You've got to work in the community. You've got to engage, you've got to listen, you've got to be respectful to people. Because respect is another life attribute that we need to have. We've got to treat every single person, doesn't matter who they are. Whether it's the president or whether it is the cleaner. Everyone deserves a right to be respected. So that's another life skill. Okay, so I'm going to pass through some of these slides quickly to allow you to ask me questions. I want to say that the late Archbishop Tutu was one of the first people from South Africa to have his entire genome sequenced. So you know, from the early 2000s, when the human genome sequence was published, there was a rush for everybody to publish. It used to cost something like 10,000 US or American dollars to do one sequence. Now, excuse me, it's much, 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 much cheaper. So some people were interested in cancer, prostate cancer, and some of you may know that late Archbishop Tutu was a prostate cancer survivor, and he had complications from that, and it was well known in the public domain. So he volunteered to have his DNA sequenced. And so he and a few other San individuals from Namibia consented to do this. So those were the first whole genome sequences in the public domain. So my former student and some collaborators from Sweden, we did some studies on some local populations, and we showed quite convincingly, if you look at this map, where the reddish part meets with the blue, the reddish part are the genetic signatures found in living San and Khoi people, almost exclusively in them. And that takes you back to over 150 to almost 200,000 years. So for that amount of time, the people we today make reference to as San and Khoi, their ancestors had some of the most ancient DNA signatures found in living people anywhere in the world. And then on the other branch, then you can see some sub-branches that we see in other parts of Africa. But when you compare this to the rest of the world is like one crunchy part of the tree. So when humans left Africa some 70, 80,000 years ago, they took with them some of the DNA that they had. So imagine a bowl of marbles, and you've got different colors, or like Smarties. Put 10 boxes of Smarties in a bowl, and then just put your hand in and see what comes out. Maybe you got more yellows and green. That is what went outside of Africa. And from that, the rest of the pool in the rest of the world's DNA is derived from. Whereas in Africa, we have this vast diversity going all the way back. And more changes have been happening over time. So, but when these early people left Africa through maybe the Egypt connecting to the Middle East, who did they encounter? Some of them encountered Neanderthals. So on a cozy night with some red wine and some candlelights, some hanky-panky was happening. And the question is, did they actually exchange genes? And for a long time, just using mitochondrial DNA, the evidence pointed to no, because there was no evidence of Neanderthal mitochondria in humans and vice versa. So when the first studies came out by this guy holding the skull, his name is Svante Pääbo. And in fact, at the end of last year, he received the Nobel Prize for Physiology and Medicine. Now, I have worked with him and with his colleague Mark Stoneking, with whom I did my postdoc, so we go back a long, long way. I've even visited his lab at Leipzig in Germany and gave talks there. But his lab mastered the art of getting DNA out of ancient DNA material, meaning material that's been lying in the soil for many, many years. In fact, Svante, for his PhD, he got DNA out of the brain of a Neanderthal mummy, from a mummy specimen. So that's when he pioneered the technology of getting DNA out of these specimens. And remember, contamination is a big, big problem when you work with these things, so you have to be careful. Anyway, mitochondrial DNA said no, but when we looked at the nuclear DNA, there was evidence of exchange from Neanderthals to humans, and something like 4% of DNA found in people outside of Africa have Neanderthal specimens. So Did they exchange genes? The answer is yes. Okay, so inclusion, colleagues. I just want to say a few things about issues related to race and genetics. At the end of the day, given that our roots come back to Africa, we are all Africans under the skin. So don't let anybody diminish your respect and your place in the world by saying that because you have more pigments in your skin and you look different to other continental people, that you should be treated any differently. In fact, you should be telling them, I know where your ancestors come from, back in Africa. So Buti, you listen to me. So when you see this kind of diversity, I want for you to understand that in terms of our genetic evolution, we have our roots in Africa. Where in Africa is debatable, but I would like to argue for somewhere in southern Africa. Because if you look at the genetics, you look at the paleontology, you look at the archaeology, and other lines of evidence, there is nowhere else in the world where the different disciplines of science converge to some root in southern Africa. So think about that. So we at the lab, when I was at Human Genetics at WITS, we offered genetic ancestry testing. People were excited about the power of DNA and wanted us to test their DNA to trace their roots. And for a documentary I did for Carte Blanche several years ago, I even had the privilege of testing the DNA of late President Nelson Mandela. And his DNA was on the branch where the star is. That is a branch that is very commonly found among living Khoi and San people. And when I gave him his results, I had the privilege of presenting his results to him. He was, oh, that is why the people say that I'm like a colored person, because my mitochondrial DNA come from the colored people. So I've had the privilege, and my colleague here, Raj, who used to help me in the lab, also we've studied over 10,000 individuals locally for ancestry tests. So even Darwin, remember we all think about Darwin as one of the fathers of understanding evolutionary theory. Well, even though Darwin is dead, his great-great-great-grandson had, Chris Darwin, had a DNA result done, a DNA test done, and we could trace his Y chromosome to that particular branch. I don't want to throw these branch names at you, but we give the the branches on the tree names. And we can trace it and see how it came out of Africa and how it eventually spread through migration and spread of people to other areas of the world. So we can get DNA from people who are not living today, and we can ask several questions about the past using DNA as a tool. And we were privileged to work with Jive Media. We created some cartoons to tell the story, because if you tell a story, it is so much nicer. So the Gogo is in some homestead in a rural area, and her brother had a DNA test. Her daughter was studying at university, and they talk about DNA. So normally, when we hear our stories from our grandparents, isn't they tell us nice stories about the past? All of that, that is called oral history. When you hear stories spoken to you, that's an oral narrative, so that's an oral history. Then there's some people who document history. That's the written history. Then there's another history that is in every single cell of your body that's waiting to be excavated using the tools of genetics. And so that's the genetic story. And at some point, each story goes only certain way into the past. So if you ask the question, what did humans eat 5,000 years ago? What system would you want to test? Anybody? Would you use genetics? Highly unlikely, right? But would you use archaeology? Remember I said settlement patterns, you can see what people were eating, the relics. Sometimes if they were eating cow, you'll see the bovine bones. If they were eating fish, you'll see the skeletons of the fish bones and stuff. So, depending on your question, you would use a particular tool. And coming to the meat of the talk, everything I was talking to you about now is a link of using your discipline and engaging with people. That's the society. But I also want for you to understand that as you grow up and you find yourself in particular niches of the workspace, don't forget that we are all humans and what humanity is. Because that's a very important thing. You can be the richest person in the world, but if you're a true, I don't want to use the politically incorrect word I wanted to use, but if you're a person that shows no respect and you forever like looking down upon people, etc., then you're not worth the money that you have. You must be caring, you must be respectful, and whatever little you have, if you can pass to help somebody else, you know that pass it over syndrome? Please try to do that. Because that is what our humanity is about. Recognizing that we are, people of the same bigger family. If you think about the earthquake that's happened in Turkey and Syria, see how the world is converging to help. Then you look on the other side, the conflict between Ukraine and Russia. So on the one side, people are helping to support from a disaster. The other side, there's conflict and war. And other people are helping one country over the other. Weird world we live in. But you as an individual have the capacity to think for yourself, to make decisions based on information you have, and when you do make a decision, make it a win-win scenario. So that's my message on life again. So as we reflect on these issues on humanity, we should try to do good wherever we can. And with that, I'd like to bring this talk to a close to say, we live in a world that has many tentacles like an octopus. We reach out in many corners. I'm a scientist by heart. I work in a space with the Academy of Science of South Africa. The Academy is the top scientists in their respective fields who get nominated and elected to become members of the Academy. There are only about 650 plus members of the Academy, which has been growing since the concept of the Academy was started about 1996, with Nelson Mandela being the chief patron at the time, and so we've grown over the years. And members of the Academy give off their time to intersect with societal activities. So this initiative, in partnership with Sci-Bono, is about science engagement. How do we engage with what our knowledge is to inspire you, to give you opportunities for careers and thinking? All I want you to do henceforth is to make informed decisions about where you want to go, what you want to do, and to be a good person. So with that, we bring science and society together, but government has the power to make decisions on policies. So we also try to engage with them to enhance their portfolios, to bring science into the conversation with academics, well, people like at universities, etc., the general public, the business sector, and with government. We call that the quadruple model. But don't worry about the model, but we need to all talk to each other so that as a collective we can contribute to the enhancement of the bettering of the lives of all peoples in this country. Thank you very much.
Thank you. Thank you for that quite profound insight. In our midst, we are joined by two schools that two different schools who are doing grade 11. And I've seen the interaction that they've been listening, which is great. I've also taken the message from what you have taught us, humanity and human, and of course, love thy neighbor as thyself, in short. I'm going to take 4 questions. I'm sure 2 from this school, another 2 from another school. Mr. Kahisho Mokoena will assist us with that. I think we also have people that join us online because this is running live as well, this talk. So without any waste of time, because I also promised management that we are only going to take 1 hour for this talk. So I can open this for now for questions, for us to ask questions to Prof. We'll raise your hand. Mr. Mokoena will come with the mic to you, and then we can facilitate questions. Let's see, any question from, because I've seen people have been listening, taking notes at the back, so I'm sure we can have some questions. I've looked at you, I've looked at your hand. There's 3 hands at the back, Mr. Mokoena. Any hands here?
Good morning, Professor. Thank you for the wonderful presentation. This is Homoto from Oshodi. And the question that I have is, what are the core ethics one should possess to succeed as a genetic scientist researcher?
Thank you very much. Should I take another question or answer one at a time?
Let's answer one at a time.
Okay. So the starting point of ethics is very, very important in genetic research. In fact, there was a project called the Human Genome Project, and that was started in like the 1990s already, where they wanted to do the sequencing of people around the world. But a key factor that kind of stalled the progress was the fact that a good ethical standard was not in place. And so that is when ethics kind of got its grounding and became into the public domain. And so the ethics is about doing no harm, being beneficial, the informed consent, and not exploiting people. So there are certain terminology that is used in the ethics jargon of how, the 5 principles of ethics. But essentially, the field of genetics, and in fact any field where human subjects are present, has to follow these guidelines. And if you're a university scientist, your university has an ethics committee. You've got to submit your application and whatever to them, they would review it. And then if you have not paid heed to some of the things for good ethical practice, they will send it back to you. If they are happy, they will give you an ethics clearance certificate that allows you to continue with your work. Because when you want to publish, the journals that will publish your article asks you for this ethical protocol. So informed consent is like the lead, but at the same time, you must do beneficial things, you must cause no harm, and you need to promote good relationships in the work that you do.
Wow, thanks. Let's get the next question.
Greetings to the house at large. I have a question about mitochondrial DNA. You did mention that it's passed down by women only. So I wanted to understand, if I'm a son and I come from a woman, obviously, my child is going to get the mitochondrial DNA from the person that I get to have a child with.
Correct.
So if you trace, you're going to trace using her DNA, her mitochondrial DNA.
Correct.
And you're tracing her track only.
Correct.
What happens to mine? How do we
You have to come to be tested.
Oh, okay.
Yeah.
So my track can also still be
Yes,
tested.
Your lineage will be what you got from your mother.
Okay.
So that's a very good question, because often people would come to our lab for testing, but they want to know something on your mother's line. But if you're testing your son, your child, he's going to get his mother's lineage.
Okay.
So we would say, no, no, no. If you want to know about your line, then you need to come to be tested.
Okay. Thank you.
Very good question.
Thank you so much.
Okay, the next question. Okay, the next question.
You see, the way the mic was passed from one to the other is like a lineage trail, just in that row. That's how mitochondria is passed along one line lineage.
Greetings all. I just wanted to ask this question. What is the Eve gene, and is it still found anywhere in the world?
Okay, the Eve
Gene.
Okay, there's no such thing as the Eve gene. So early on, when mitochondrial DNA was first published in 1986, in a groundbreaking study that was published in Nature, the writers called it mitochondrial Eve, the point of connection of the most recent common ancestor for those lineages they call mitochondrial Eve. Now, in the Bible, they talk about Eve and Adam and Eve, so this created a lot of conflict because that point was over 200,000 years. So there is no gene per se that is related to Eve. So the Eve story is in the biblical context. We prefer to say the most recent common ancestor. Now, the point I want to add, and thank you for asking this question because I don't have a slide for it. When we connect, if you think, imagine a tree, right? You have many leaves on the tree. You can all see the leaves all over. The leaves are connected by the twigs. They come into smaller branches, then bigger branches, and then you get to the common trunk. Now that's how we trace DNA. We can put all of you in the room, and I can identify your leaves, and I can show how we are all connected. But when you go to the point where all the lineages connect to the common trunk, there were other women who lived in that generation, okay? But their lineages went extinct. Can you tell me or think about what would cause lineages to go extinct? Anybody? So, when I say we connect all the lineages to a common ancestor, that's what has survived. But at that point, back in time, 200,000 years, there were other women who lived, but their lineages didn't make it to the present, it went extinct. Which scenarios will contribute to some lineages going extinct? Only having sons? The sons don't pass it on? A major catastrophe, say like an ice age happened and wiped out one particular line in one area, extinctions due to catastrophes. Sometimes the person carrying a unique line may not have produced any offspring, least of all girl children. Okay? So the point of most recent common ancestry did have other women, but those other women's DNA got lost over the years that it didn't make it to the present. Okay? That's the answer I offer to your question.
Special greeting to you all, and special greeting to you, Professor. What I would like to ask you is that, can you please Can you please enumerate on the significance of chloroplastic DNA?
Of?
Chloroplast.
Chloroplast.
Yes.
Oh, chloroplasts are in plants.
Chloroplastic DNA.
Chloroplastic. I'm not sure I understand. Chloroplastic. I'm sorry, you got me there. You want to help me? Chloroplastic. Raj?
You needed some elaboration on that. It appears, I'm sure that it's one of the slides that you have touched upon. So I think we wanted you to elaborate on that. The chloroplastic DNA.
I don't have a slide with
It wasn't
chloroplast.
chloroplast. Perhaps there was a chloroplast.
Okay. I'm sorry, you got me. I don't recall. I know chloroplasts and stuff like plants, but I'm not talking about plants. The girls, y'all are losing, huh? You're letting me down. No girls are asking me. Yes, there's one.
Good day, ladies and gentlemen. I wanted to ask, let's say we clone a human being or a plant. Will it have the same DNA?
Very, very good question. Cloning is the way of making an exact replica or copy of what you have. Okay? So, some time ago, the first knowledge about cloning was with Dolly the sheep. So, many of you may remember, you're too small to know that. But what happened was, they cloned some cells from a sheep. So, they took some of a cell line of breast tissue and they had a cell. Now the cell has the nucleus. So what they did was, they took another cell, took out its nucleus from the recipient, okay, from another specimen, and they sucked out the nucleus from the donor and injected it into the cell without the nucleus. And they hit it with some vigorous electrical pulses, so much so that that cell started to grow its own cell line. Okay? But once that started, they implanted it in a surrogate sheep that carried it. So that was now taking a copy of a donor and putting it into a replica. But when you do this cloning, the mitochondria of the recipient cell is not the same as the donor. It's only taking the nucleus from the donor and sticking it into the recipient. But the recipient's cytoplasm has all the other cellular content. And so that is a clone. Okay? A clone is an identical copy of the nuclear DNA in the recipient. The question that then becomes necessary is what the first gentleman asked about the ethics. The fact that we can do it, should we do it? And recently there was a huge debate in the literature about the possibility of cloning of humans in China. The scientist apparently cloned and he was struck off the register and there was a whole debacle in the media about it. In fact, the National Academy of Science in the US hosted a big meeting. I attended that meeting as well, representing our academy. And so there's a freeze on germline cloning. Anything to do with reproductive cloning is banned in humans. It does happen in some animal species, but if you take a somatic cell, like you take a skin cell thing, and you want to create some tissue that could be useful for some other usage, that is allowed. So again, I'm going to use some jargon terminology. Somatic cell cloning is allowed, that is other cells, but not germline or reproductive cloning. In humans, that's not allowed. So while it is technically possible to do these things, it's not encouraged. Then there's another question that I got asked for a radio interview. Like, you know, the woolly mammoth that's gone extinct. They found some specimens in a frozen environment. So it is possible. You may see movies about freezing people and they come back later and so on. That's called cryogenic. Freezing is cryogenic. So the question to me was, given that we have these new techniques in genetics, do you think we should resurrect the mammoth? Like if you watch Jurassic Park, the way they did it with the dinosaurs, should we bring back dinosaurs? There you go, you people. We need to do a course in ethics. So what happens is we have different environment, and there's another science of biology called conservation science. You know, we are part of a macro environment. We live as humans, Homo sapiens sapiens, but we have a world of floral diversity and faunal animal diversity, plant and animals. And over the years, things go extinct. Global change, climate change, and all of that is contributing to changes in our macro environment. So now we bring something in that's been extinct for so long into the contemporary, in the present. We are going to be tampering with that environment, the conservation space of these species. So it becomes a problem. It becomes a problem, and so scientists debate. Some people are pro, and others are anti-human intervention in certain instances. So like our rhinos being poached. So what do we do to mediate? We catch the rhinos and we cut off their horns. So we give them an opportunity to live, but they're living without their horns. Their horns evolved for a purpose. Okay, so now they can't go snouting and doing things because they don't have a digging tool. But at least, rather than them going extinct by being killed by poachers, we give them a chance to live, but they may not have the full life that they would have lived if they had a horn. And I'm not saying that that is a significant change, but you have to make a balancing act. Life versus extinction. So these are things, and I'm glad it comes up, because these are things we've got to constantly ask ourselves. And the ability of us humans to think and the capacity to stretch our thinking is a trump card on the one hand, but also a threat on the other hand, because sometimes we overthink, but for the wrong things. Cool.
Thank
Yes?
you, Prof. I think that will be the last question that we are taking.
There's one here. Stand up and shout your question.
Greetings to all.
She's going to shout like me.
Greetings to all of you. Can I ask, where do you find the mitochondrial DNA of the past to match today's mitochondrial DNA to know a person's lineage?
Okay, where do we find the mitochondrial DNA in living peoples to connect it to the past? So basically, we don't know. We don't have a sample from the past. Okay. So what we're doing is we're studying living peoples, and we reconnect how they come together to some point that could be an ancestral one. Okay? To give you an example, if your kitchen is tiled, right, and you're standing on one tile, you can go one step at a time in many steps to get to another point. So what we do, we use mathematical tools to reconstruct the best fit of connecting all the patterns we have today to a common point in the past. So we don't have a living person in the past, we infer what that ancestral lineage would have looked like. Okay?
Greetings to all. My name is Kamahelo Tolo from Himla Secondary School. The question I have, ma'am, is genetics linked to how people age? For example, you have people like Steve Harvey, who is in his 60s, but he looks like my dad who's in his 40s.
Yes, no. There's a whole science on aging. So in the mitochondria also has a role in playing in aging. So this is not the evolutionary thing, but some of its other properties. So we have these free radicals that occur in the structure of the mitochondria. So some of these free radicals, etc., or the oxidative property of these energy molecules do contribute to the extent in which we age. But we don't have all the answers. So, the normal thing is they say, well, depending on the genes you've inherited from your parents before, you will see what your longevity would be. So that's one answer. There's another thing related to, and this is a complicated response. We have methylation of cells happening. So some of these methylation activities contributes to how long we would live. So you have people in Japan where you have many, many people over the age of 100. They call them centenarians. And when they studied some of their genetic properties, they discovered certain methylation properties. And then they took another sample from Scotland and they simulated this based on what they learned from the Japanese, and they found that they could predict at which age these people will even die. So there's a big science out there. It's not my specific area of research, but there are studies ongoing about these sorts of things. So that's a very good question, too.
Thank you, Prof. I think from our management side, we can also just get questions from there as well.
Thank you, Prof. A common thing which happens in our families is when a child is born, and the grandparents would say, he has this one's nose, he has this one's ears, the eyes are green. No one ever had green eyes in the family. So can you, in simple terms, explain paternity testing?
Yeah.
Which is a common thing nowadays.
Right. So paternity testing and what you acquire through inheritance are slightly different things. So sometimes you'll say, somebody say, oh, you got a temper just like Grand Auntie Daphne. Or, so we see traits of behavior in other people. Whether that trait is genetic or not is debatable. So why some of us may have green eyes, it all depends. Remember we talked about Mendelian inheritance, about dominant and so on. I didn't go into that in detail. But these are things, there's certain traits that are dominant. If you have a dominant trait, you will express that, what we call the phenotype or the visual of that particular trait. If it is recessive, then it's a luck of the draw, so to speak. But when it comes to paternity test, that's a different scenario. So if a child is born, and there's a question of whether the father is the biological father or not, there are tests that can be done. And in fact, my former department of human genetics does that. We also got the testing to do for people applying for residence in South Africa that require the paternity or parentage test. So what happens is, you have the DNA of the child, you have the DNA of the mother, you have the DNA of male 1, and if let's just say there isn't any other male, this father wants to know, is this my child? So you go to the DNA testing facility, the nurse will ask you for particular information, check your ID, all of that, take copies of it, and they will take a blood sample, a or the cheek swab, depending on what they use. And then we have a kit. It is on the nuclear DNA. And that kit has about 20 different genetic systems that we test for. Then we test the mother, we test the father, and we test the child. Remember I said mother gives a pair of chromosomes, one will come from her, and father will give one chromosome of his two. Now we know the pattern that the child has inherited. Now we will see. Is it found in the mother? Usually you'll find that in the mother. As you know me, the mother is the constant. We very, very rarely, except when babies are swapped in hospitals and stuff, we have these stories where there's a non-maternity. So once you identify the mother's line in the offspring, now you check this other chromosome that's in this boy or in the child, does the father have one or the other of his chromosomes to match? If it is not, then he can't be the father. So that will be a non-paternity. But the courts don't accept that exclusively. You then have to give them a mathematical, statistical support for your test. So then the lab goes and matches, because sometimes it may be the same at about 19 of the 20 positions, but one may be slightly off. It could be a mutation from one generation to the other. But they will work out statistically how often that happens. And then they'll give you a probability of a match, 99.9999% or whatever the case. I won't always say 100%, but that's how we do it. If one of the chromosomes in the child is found in the male that was tested, that we will say matches, is the father. But now let's say two men came and they said, that child is mine. We also take a sample from the second male, and we test both males and to see in which of the two males tested that paternal chromosome can be identified. And that's how we do the paternity testing. So it's doable, and it happens. We get cases where there are people contesting wills, all kinds of scenarios for testing. And even Home Affairs requires testing of children who are claimed to have been fathered by a South African male for residency and so on. So it's very complex, but it's doable.
Is it exactly the same test if swab kits for the mother?
Swab kits.
Let's say hospital.
Yes. A swab is taken to get a sample.
What I mean,
Swab. Yes, exactly. But if it's a non-maternity, we'll do the same. We'll test the child, and we'll test both women. And then now we will try to match, because you will be tracking the mother's chromosome. There will only be one that will match usually, unless they are twins ahead of the game who will have identical chromosomes. Sometimes siblings, like an uncle who has raped a child, because some of the chromosomes may come up. So the lab has a way of tracking all of that from the pedigree.
Thank you very much. I know colleagues have been participating, and you can see from the house that there are so many questions. And they will get that opportunity to ask you questions as soon as we finish with our program. I think you'll still be around 5 minutes that you can spend with them, because I can see the topic is quite engaging. And that is very much nice if the topic is like that. Thanks. Before you sit down, you spoke of one of the values that is very much important. You spoke of respect. And I've taken that into consideration. Thank you very much. But even here at Sci-Bono, we also even reinforce even values of gratitude as well. And we have a way of doing it, because it has to be in public. We need to show everybody that we have gratitude. But just to highlight something to you, why I also invited you as we're engaging in our conversation. I happen to have one of the gifts during the year as a book. And this book, the title of it is Freedom. It's written by an Australian biologist, Jeremy Griffith. When I was reading it during this December, it explained exactly what you have just said here. And surely there is trouble and humanity is needed. With these words, I shall now call upon our Career Center Senior Manager, Ms. Felicia Segele, to come and provide us with vote of thanks and give gratitude to our professor in blessing us this year. Ms. Felicia Segele, can you please come to the stage? Thank you very much. You want okay.
Is it working? Thank you. Good morning to you all. Good morning to all the boys and girls in the audience. Good morning to Prof. We are delighted as Sci-Bono to have you here. It is a seminal moment for us here because we've been wanting you to come to Sci-Bono as far back as when you were still with the NHLS. Today is that day. It's a seminal moment. And thank you so much. I'd like to thank you specifically because I think you are a beacon of hope for all these boys and girls in the audience who maybe would otherwise not have aspired to become scientists. I know that you've also modeled to them what it could be to be an accomplished citizen. I mean, you've come a long way, and I'm sure it wasn't easy as a woman scientist, and I'm sure the young girls in the audience, as well as the boys, will learn something from you. For me, personally, I thank you for reminding us of the ethos of humanity and humility. It is something that we all need as a society, and to all the boys and girls, as you one day become doctors and professors like Professor Soodyall is today, please take that with you all the time. It's at the core of our being, of who we are, and of who we could become if we really uphold those values. Again, to the boys and girls, I was impressed. I am impressed by the level of questions that came from the floor. It just says that you have an interest in things science. And I'm hoping that this conversation today with Professor Soodyall has piqued the curiosity in you enough for you to go out there and want to study science. Because any society, any community is as strong, as safe, as secure as its knowledge, scientific base. And we look to you for that. And we thank you for making the time. Prof. Soodyall, as a token of appreciation for us at Sci-Bono, that we are blessed to have you today. Finally, I'd like to present this to you. Yes, it is. You are an epitome of a servanthood professorship, because that's what you do. You pass the baton and inspire the young ones, so that long after you're not here, we still have other scientists. Thank you so much. Thank you.
Thank you, Ms. Felicia Segele. Ladies and gentlemen, I think we've taken too much time because we were supposed to be leaving the auditorium at 12 o'clock. But because of the interaction, we had at least to make sure that we can accommodate at least everybody to understand one another and to hear from others what is their point of view. From me, Tolani Nameka, from our team at Sci-Bono, to the guests that have joined us in this auditorium and those online, to the IT people who have made this possible, to our bookings team who have made it possible for you to be here as learners. I'm saying to them, thank you very much. Let it not be the end for today. Let's get more from them. Thank you very much, ladies and gentlemen. Thank you, if you can take them.